Assalamualaikum and hello everyone. My name is Dayana Nufarhana, S46915. Today I would like to share about our journal, that is monosex production of African catfish, Heterobranchus longifilis through Gynogenesis and Androgenesis. As we know that biotechnology can be defined as the use of biological processes for the improvement of the characteristic of economically important plants and animals. This has been a tool for the improvement of fish stock employing chromosome manipulation techniques, leading to desirable qualities. The techniques more commonly employed include gynogenesis. So I will tell you the definition of gynogenesis. It is involve diplodization of the maternal genome through polar body rotation due to activation of an egg by a sperm that degenerates without fusing egg nucleus. And it is a process to produce individuals from maternal chromosome only eventually to obtain homozygosity. Next is to form inbred lines to achieve proper hybridization and selective breeding. It is fertilized with irradiated sperm. Most of the more recent protocols for gynogenesis have used ultraviolet irradiation to inactivate paternal genome and temperature shock to restore the diplody. So the product, if the female is the homogenetic sex, all female lines can be produced by gynogenesis in one generation. So I will pass to my friend for explain more. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. My name is Rabiatul Adawiyah. Today I would like to share with you about gynogenesis process. The gynogenesis is defined as the process of development of ovum after penetration of sperm, but in the process sperm nucleus is unable to form sagami with nucleus or ovum. So gynogenetic fish inherit genes only from their mother. The penetration of sperm is responsible for initiation of development alone. Gynogenetic haploid embryos contain a haploid chromosome complement representing a single product of a meiotic division. 1/4 of a tetrad embryo do not develop entirely normally. This picture shows how chromosome set manipulation induction of gynogenesis in fish. Gynogenetic diploid embryo contain a diploid chromosome complement, also derived from mother only, and are obtained by treatment to prevent the second division in an egg fertilized with UV irradiated sperm. In fish, the gynogenesis is achieved both naturally and artificially. In some fish, gynogenesis is a natural mode of reproduction. These unisex fish may be often diploid. In a few fishes, such as silver crucian carp and pichiliocyp, cis unisex form present in natural population. For artificial gynogenesis, can also be produced under suitable experimental conditions. During artificial gynogenesis, manipulation in event of fertilization are made by destroyed DNA of the spermatozoa and restores diploidy to the ovum with either meiosis or cell cleavage. In the process, the female inheritance is included. The offspring obtained in this way process inheritance of female character, and there are only one sex, that is female.
Gynogenesis, the stripping, activation, hatching, survival monitoring and feeding step are similar to the androgenesis experiment, except for the sperm was irradiated. The irradiated sperm was used to fertilize normal eggs, and the zygotes were cold shocked at 50 Celsius for 15 minutes starting and 40 minutes after fertilization.
occurs hatching and survival rate in embryo of irradiated sperm and not irradiated egg without full shock haploid we use a gynogenesis control. From the table, without using full shock, number of hatching is 98%, percentage hatchability 64.8, survival at day 2.0%. Second, hatching and survival rate in embryo of irradiated sperm and non-irradiated eggs with full shock, which is gynogenesis. Number of hatching and percentage of hatchability of gynogenesis is lower compared to haploid, but the survival rate of embryo of gynogenesis is increased
Hello, my name is Siti Nushahira and I will discuss on the result. First point is the level of hatching between gynogenesis and androgenesis. The gynogenesis after the diploidy restoration, also known as the Cook shock treatment, produced heterozygosity. Meanwhile, androgen after the diploidy restoration produced homozygosity. It has been studied that the heterozygosity have higher reproductive success than the homozygosity. This have been result in higher survival rate in Gynogen method. The figure here we explain the meaning of heterozygosity and the homozygous. The heterozygous have two different alleles, which one is dominant and the other is recessive, while the homozygous have both dominant and both recessive. A heterozygote has advantages due to a single locus known as the overdominance. The next point is high gynogenesis progeny. Gynogenetic diploid were due to a meiotic crossing over in the region between gene and centromere. This eventually determined the rapidity of production line. This figure shows you the meiotic gynogenesis and the androgenesis process.
Assalamualaikum. My name is Nurfati Izzati and I will talk about other point of discussion. The first point is there are low or zero amount of androgen were produced. Based on the study, hatching and survival difficulties of defertilized egg occurs because zygote are diploidized by a late shock, causing all the loci to be homozygous, which will lead into little genes. And even zygote that survive, it may have reduced in viability because of genomic influence. Next, androgen is difficult to produce than gynogens. This is because androgen are totally homozygous, which is present to be large amount of noxious genotype. Last but not least, the uses of UV radiation in this study. UV radiation is widely used because it is easy and safe to apply. It gives less residual paternal inheritance and also have low genetic inactivation potential on egg. That's all from us. Thank you.
Hi, Assalamualaikum. My name is Fatih Amira binti Muhammad Sure. So I want to conclude. These experiments carried out on African catfish, Heterobranchus longifilis, have proved that it is possible to obtain gynogenetic progeny of fish. Investigations have revealed the cytological mechanics and genetic characteristics under radiative diploid gynogenesis. The aim of future experiment is to work out method of raising the numerical strength of diploid gynogenetic offspring. It is possible to visualize the prospects of using artificial diploid gynogenesis. This method holds great prospects for intensifying homozygotization in some line of breeding. Within one generation, it is possible to obtain a result that otherwise require a long period of inbreeding, which due to late maturation of African catfish is very impeded. This method undoubtedly will be useful in selection of heterosis combinations. Such heterosis will evidently be possible in crossing of the top cross type. Thank you.